Hey everyone, this is Miss Moriarty and I'm here to discuss with us topic 8.8, which is all about bioaccumulation and biomagnification. So how do chemicals wind up accumulating in organisms? Well, right, we've been talking a lot recently about chemical compounds that tend to persist in the environment for long periods of time, tend to be fat soluble, and when they are fat soluble, if an organism were to consume, say, another that's been contaminated with something like a persistent organic pollutant or a compound known as methylmercury, what's going to happen is it's not going to easily dissolve in water or their blood. What it will do is dissolve in their fat tissues and accumulate in those tissues over time. So that's where we get that term bioaccumulation. So we see an increase in concentration of some sort of pollutant or chemical compound in an organism over time. But then what is biomagnification, and what is the difference between the two? So here, biomagnification is when we see an increase in a chemical concentration in animal tissues as you move up a food chain. So increasing concentrations of those types of fat-soluble compounds are going to increase as you move up in trophic level. Now, a part of the reason why that is goes back to Unit 1 topics when we talked about the energy pyramid and the 10% rule. We know that at the base of the pyramid are going to be producers. So that could be like phytoplankton or grass or really any kind of vegetation that may be found in that ecosystem. Persistent organic pollutants or other types of pollutants like methylmercury can be found in sediment or in water, and that's how that vegetation can become exposed to it. Primary consumers, maybe like bottom-feeding fish, insects like grasshoppers, zooplankton, are going to consume the producers that are now contaminated with that chemical compound. It is then going to bioaccumulate inside of their tissues. But when a secondary consumer then eats the primary consumer, they take in the POPs in that organism's tissues. And because of that 10% rule, we know that that pyramid, as we move up in trophic level, gets smaller and smaller. Less available energy, which means you're going to have to consume more biomass in order just to survive if you're an organism who is found at the very top. So this leads to higher and higher levels of those chemical compounds in their lifetimes. So large predators like a salmon, a dolphin, a whale, or even a human have some of the highest amounts of persistent organic pollutants or methylmercury found in their blood. Now, some examples of biomagnification. A classic example was actually the biomagnification of the pesticide known as DDT. DDT was used to eliminate mosquitoes. It's been phased out in the United States at this point. However, in other parts of the world, they actually still use DDT to try to rid the area of mosquitoes. But what they found in the United States when we were using DDT to eliminate mosquitoes, we found that at the highest levels of the food chain, like our eagles and our osprey, they had the greatest amounts of DDT concentrations compared to really any other point in the food chain. And as a result, this caused thinning of the eggshells of those eagles and ospreys, which led to a massive decline in population because they were no longer to properly reproduce. So this kind of prompted what's known as the Endangered Species Act and obviously the phasing out of DDT. Once we started to see this, we saw then the populations of especially eagles kind of kick back up. Another example of biomagnification is when we see the compound of mercury being emitted into the air. Where could mercury come from? Anthropogenically, it can come from the burning of coal. Naturally, it could come from a volcanic eruption. It can be carried by wind, deposited in water, where bacteria in the water are going to convert it to a toxic form known as methylmercury. Methylmercury can then be taken in by phytoplankton and biomagnification magnified at much higher trophic levels. So things like tuna, sharks, and whales can have higher concentrations of methylmercury as opposed to phytoplankton or maybe even zooplankton. When exposed to methylmercury, it can act like a neurotoxicant, and we know that that prefix neuro, that refers to nervous system. So when you have a high exposure of methylmercury, over time that can cause nerve damage. Now, human exposure to methylmercury and persistent organic pollutants can happen for us too, especially with people who heavily rely on seafood as a part of their diet. So eating large predatory fish like tuna and salmon does put us at a greater risk of exposure to mercury than if we maybe eat a little bit lower on the food chain. People who consume high amounts of fish that can potentially have mercury in it can damage their human nerve system, especially to developing fetuses. So women who are pregnant should stay away from eating types of seafoods. Can also disrupt reproductive systems and impact fertility. So here's our practice FRQ for 8.8. If you want to pause the video at this point, take a moment and answer the practice FRQ in your notes and on the Edpuzzle. Okay, at this point we should have paused and answered our FRQ. So here our FRQ states that scientists are interested in determining if selenium from a nearby mine magnifies in the tissues of fish living in a nearby lake. The first part of the question asks you to describe the difference between biomagnification, bioaccumulation, and identify what a potential testable hypothesis for this study could be. Well, remember, bioaccumulation is when chemicals like DDT or selenium in this case can build up in the fatty tissues of organisms because they don't easily break down. But biomagnification states that when animals eat other animals, we see a magnification or a higher concentration of that pollutant found in the food chain at higher points of the food chain. A testable hypothesis in this case could state if selenium is present in the ecosystem, then it will be at higher concentrations in fish tissues rather than in the tissue of fish prey. Because recall, pollutants like our selenium, DDT, PCBs, PFAS, all of those have the potential to accumulate and magnify at higher trophic levels than they do at lower trophic levels. All right, everyone, that's it for our video on 8.8. Please leave a question.